Hi, my name is Pumika, and in this chapter, we're going to be studying about the transmission of the nerve impulse. We've already done the structure of the neuron and the various types of neuron and how they pass messages. We're now going to do how a nerve impulse it travels down the axon. Starting out, the transmission of a nerve impulse along a neuron from one end to the other end occurs as a result of electrical changes across the membrane of the neuron. The membrane of an unstimulated neuron is polarized. That means there's a difference of charge between the outside and the inside of the membrane. The inside is negative with respect to outside. So you got to remember this. The inside of the axonal membrane is negative. Polarization is established by maintaining an excess of sodium ions on the outside and an excess of potassium ions on the inside. A certain amount of Na plus and K plus is always leaking across the membrane through the leakage channels, but Na plus K plus pumps in the membrane actively restore the ions to appropriate sites. So that is how it happens, the process of polarization, then depolarization, then repolarization. So there are certain pumps which are present which kind of keep supplying Na and K plus ions onto both the sides wherever the requirement needs. The main contribution to the resting membrane potential, which is the difference in permeability of the resting membrane to potassium ions versus the sodium ions. The resting membrane is much more permeable to potassium ions than to sodium ions, resulting in slightly more net potassium diffusion from the inside of the neuron to the outside. So you got to know this. The permeability is more to the potassium ions, and some amount of potassium ions always keep leaking towards the outside, and potassium is by convention present inside. So we have then the sodium ion diffusion from the outside of the neuron to the inside, causing the slight difference in polarity right along the membrane. Polarity right along the membrane of the axon. Other ions such as large negatively charged proteins and nucleic acid reside within the cell. It is these large negatively charged ions that contribute to the overall negative charge in the inside of the cell membrane as compared to the outside. In addition to crossing the membrane through leakage channels, ions may also cross through gated channels. Gated channels open in response to neurotransmitters, changes in the membrane potential, or other stimuli. So we've got two types of channels, the leakage channels and the gated channels. The leakage channels are usually open, and the gated channels are usually voltage gated channels, which open or close according to the stimulus that is given to the neuron. The following events characterize the transmission of a nerve impulse. So we're starting out. Firstly, we have the resting potential. The resting potential describes the unstimulated polarized state of a neuron. So that's the resting potential, which is about minus 70 millivolts. You can also represent millivolts by MV, a small m and a capital V. The graded potential. A graded potential is a change at the resting potential of the plasma membrane in response to a stimulus. A graded potential occurs when the stimulus causes the Na plus or K plus gated channels to open. Then, if Na plus channels open, positive sodium ions enter, because sodium ions, as we study, they're present outside the membrane. So as these channels they open, the sodium it comes inside, and the membrane depolarizes. That is, it becomes more positive, because we knew that the inside membrane is negative. So now that Na plus has entered, that means it becomes more positive. If the stimulus opens K plus channels, then positive K plus ions exit across the membrane, and the membrane hyperpolarizes. That means it becomes more negative. A graded potential is a local event that does not travel far from its origin. Graded potentials occur in cell bodies and dendrites. Light, heat, mechanical pressure, and chemicals such as neurotransmitters are examples of the stimuli that may generate a graded potential, and it depends upon the neuron. Now, the action potential. What do you mean by an action potential? We studied that a graded potential is localized. Unlike a graded potential, an action potential is capable of traveling long distances. So this is basically it is the functional unit or the constituent unit of how a signal goes back to the spinal cord, and then it goes to the brain for analysis, and then the brain, it tells the spinal cord, and then the motor neurons go to their respective muscles, and that is how the response that the body produces takes place. So it's only possible because the action potentials are capable of moving on to the long distances. If a depolarizing graded potential is sufficiently large, Na plus channels in the trigger zone, they open. Alright, the Na plus channels open. In the response, Na plus on the outside of the membrane, it becomes depolarized. If the stimulus is strong enough, that is, if it's above a certain threshold level, additional sodium gates will open, increasing the flow of Na plus even more, causing an action potential or complete depolarization. And it's about minus 70 to about plus 30. So we knew that the resting potential is about minus 70, so it almost goes to plus 30 millivolts. This in turn stimulates the neighboring sodium gates farther down the axon to open. In this manner, the action potential travels down the length of the axon as opened. Na plus gates stimulate neighboring Na plus gates to open. So as such, imagine an axon. In the first part, the Na plus, it goes in, but because the stimulus is above a threshold value, it triggers the opening of the neighboring gated channels of sodium, and then because those open and the threshold value is more and so on, it just keeps on triggering the opening of the sodium gated channels throughout the length of the axon. The action potential is an all or nothing event. When the stimulus fails to produce depolarization that exceeds the threshold value, no action potential results. But when threshold potential is exceeded, complete depolarization occurs. Now, what do you mean by repolarization? In response to the inflow of Na plus, K plus channels will open, this time allowing K plus from the inside to rush outside of the cell. The movement of K plus outside of the cell causes repolarization by restoring the original membrane polarization. So that is how the charge kind of balances. Unlike the resting potential, however, in repolarization, the K plus are on outside. Resting potential, we studied that Na plus were on the outside, but in repolarization, K plus are on the outside to balance the charge. And Na plus are onto the inside. Soon after the K plus gates open, the Na plus gates, they close. What do we mean by hyperpolarization? Now, by the time the K plus channels close, more K plus ions have moved out of the cell than actually necessary to establish the original polarized potential. Thus, the membrane becomes hyperpolarized, which is about minus 80 millivolts. Then we have the refractory period. With the passage of the action potential, the cell membrane is an unusual state of affairs. The membrane is polarized, but Na plus and K plus are on the wrong sides, like wrong side according to the convention. During this refractory period, the axon will not respond to a new stimulus. To re-establish the original distribution of these ions, the Na plus and K plus, they are again returned to their original positions to the resting potential location by Na plus and K plus pumps in the cell membrane. Once these ions are completely returned to the resting potential location, the neuron is ready for another stimulus. So this was all about how a nerve impulse is passed out. Very easy to understand, I must say. Let's consider this graph right here. We had minus 70, which was the resting potential, and approximately minus 60 or something, closer to 45 or something, it is the threshold potential. So this much should be the value of the threshold stimulus in order to trigger an action potential. So this goes on, and here depolarization occurs nearly at 30 millivolts plus 30 millivolts right here, and then the action potential, it goes on, and here almost the depolarization occurs in the opposite direction, of course, because this is coming down, and then it reaches the resting potential, and sometimes hyperpolarization also occurs, which goes down to minus 80. So this, on the x-axis we have the time in milliseconds, and on the y-axis we have the membrane potential. So as you can see, in almost 5 milliseconds, an action potential travels down the axon. So you may imagine the amount of time that it takes for an impulse to pass across a neuron. It's so very small, and that is how we are able to respond to everything that has happened externally so quickly. Okay, in a nutshell, I'll be explaining this again. Diagrammatically, this is the resting potential. That means Na plus is outside and K plus is inside, and that is how the inside membrane is a little bit negative in charge because K plus is lesser inside. Then we have depolarization, in which the voltage gated Na plus channels, they open, and sodium, it kind of flows inside, and it kind of gives the inside membrane a little bit of a positive charge. Then repolarization begins, in which the voltage gated K plus channels open, and as such K plus, it goes outside, and because they are on the wrong sides of the membrane, this stage is known as repolarization, and it is different than resting potential because in such a case the K plus is outside and Na plus is inside, which is not the case in resting potential, because in resting potential Na plus is outside and K plus is inside. Then finally we have the resting potential again, in which both Na plus and K plus, they return back to their original position, original places, outside and inside, wherever they were, and this is also known as the refractory period. The graph is here again. You may totally consider this because this is sometimes asked in the examinations. I hope I made myself clear in this lesson of transmission of nerve impulse. If you want to watch this lesson in Hindi for better understanding, no forcing of course, you can check out my other course. Thank you so much for watching. If you have any queries, you can write it down in the Q&A section. Please follow me, rate this course, rate this chapter, and review and recommend this wherever you think you should. Thank you.